So just a word about stem cell mobilization. I mentioned earlier that stem cells can be mobilized by osteoclasts releasing a protease, and this happens naturally. But we can do this clinically as well in order to collect stem cells from peripheral blood. And this has clinical applications for growing stem cells and getting them to differentiate into different populations at need. So let's talk about some of the hematopoietic lineages. What is a lineage defined? A lineage is a family of cells derived from an immature progenitor that leads to a mature end-stage cell. So the hematopoietic lineages are the myeloid, which are all of the cells essentially except for the lymphoid ones in blood. So that would include the erythroid, the granulocytic, the monocytic, and the megakaryocytic. So all of those are the myeloid cell lines. And then there's the lymphoid cell lines, and those all lead to the various lymphocyte subtypes. So colonies were discovered in irradiated mice that were injected with bone marrow hematopoietic cells after irradiation. After several days, the spleen was found to have nodules of cells within it. These are the colonies of cells derived from the stem cells that settled in the spleen, which is a friendly environment which they can grow in. Different colonies contain different combinations of cells reflecting the different lineages of hematopoietic cells. By definition, a single cell that has the capacity to divide and give rise to a colony of cells is called a colony-forming unit, or CFU. It has been possible to grow these colonies in cell culture, which has helped to identify the hematopoietic growth factors that stimulate the growth of different hematopoietic lineages. There are many different CFUs, and we're going to take a look at a lineage diagram in a couple of minutes and see all of those different colony-forming units that lead to the end cells. So what are these hematopoietic growth factors? Well, specifically, they're proteins produced by several different organs. One example would be in the liver. They have a varying degree of specificity for the stimulation of cells in different hematopoietic lineages. They can alter normal mature blood cell activities, and they are characterized first in those tissue culture experiments which I described earlier, and some of them have been developed into very profitable drugs. This figure illustrates some of the hematopoietic lineages with the addition of some of the growth factors that stimulate the development of them at various stages. So you don't have to learn all of these growth factors, but some of the most important ones are IL-3 and SCF, which play an important role in development of multiple lineages. That's in the early stage. Erythropoietin, which is made in the kidney and is a major regulator of erythrocyte development. Thrombopoietin, made in the liver, is a major regulator of megakaryocyte development. And GCSF and GM-CSF are major regulators of granulocyte and monocyte development. So those are the ones I think that you should be the most familiar with. They're outlined in red there, and you can see which cells they directly lead to. So the real picture of this whole process is much more complicated, and there's many more growth factors. I just wanted to show this to you to make you aware that the picture is much bigger and more complex than I've shown you before. But you don't need to know this. This is just an extra FYI. So let's start talking about the specific process of some of these different lineages, starting with erythropoiesis. So the primary regulator of erythropoiesis is erythropoietin, which is made in the kidney. Production increases if red blood cell number decreases. Erythropoietin, with other factors, stimulates formation of the BFU-E precursor and the CFU-E precursor, and then eventually pro-erythroblasts. Pro-erythroblasts and the subsequent progeny down the lineage are assisted in development by macrophages, which produce yet more growth factors and also phagocytize the nuclei during development. This whole process is characterized by gradual accumulation of hemoglobin, decrease in cell size, loss of organelles, and eventual extrusion and phagocytosis of the nucleus. So let's go through this in sequence. You start with the pro-erythroblast. For now, don't worry about what that looks like. It's a progenitor cell committed to further maturation in the erythroblast lineage, and you don't need to ID it. So it progresses to the first stage that you do have to ID, and that's the basophilic erythroblast. This is a fairly small cell with a circular nucleus and blue, deep blue usually, basophilic cytoplasm. It's similar in appearance to a lymphocyte, but the cytoplasm is more intensely blue, and it has what we call a kind of a maroon red looking very round nucleus. Why is the cytoplasm blue in this cell? Well, there's a lot of production of hemoglobin going on. Therefore, there's a lot of RER in the cytoplasm, which is why it's so deeply blue. Now, as this process continues, you move on to the next stage of development, which is the polychromatic erythroblast. There is no further cell division after this stage. This cell has a more condensed nucleus than the basophilic erythroblast, sometimes referred to as a checkerboard appearance or a stippled appearance, because it has areas that are highly condensed or highly heterochromatic. The cytoplasm here is kind of half blue, half pink. Because of the hemoglobin that's beginning to build up, you have the acetophilic or pink color, and the remaining RER that's still producing hemoglobin is out there, so you have the blue patches as well. So it can be very patchy, or sometimes it can have almost a grayish appearance. Just depends on the prep. The next stage, as this process continues, the nucleus gets yet smaller and more condensed and very dark, and that's the orthochromatophilic erythroblast, or orthochromatic erythroblast. You can abbreviate it if you like that. In this stage, the cytoplasm is very pink and acetophilic. It's filled with hemoglobin at this point, and the RER has been kind of inactivated. This stage is characterized by the extrusion of the nucleus, which is then phagocytized by nearby macrophages. At this point in its maturation, it's usually released into circulation as what we call a reticulocyte. It still has a little bit of RER in the cytoplasm, which can't really be seen without special stains. So it looks just like a mature RBC, and it finishes its maturation during circulation and functions for about 120 days at that point. So let's move on to megakaryopoiesis. The primary regulator of this process is thrombopoietin, which is made in the liver. This is bound to platelets, and if a platelet count decreases, unbound thrombopoietin increases. Thrombopoietin may stimulate all levels of megakaryocytopoiesis. So these are circled cells here are examples of megakaryocytes in the bone marrow stroma, and usually you will find them sitting right next to a sinusoid because that's where they generate their platelets, right out into the sinusoids. So here's the ultrastructure of them. They have a multi-lobed nucleus, and they have a cytoplasm which is filled with these demarcation membranes, and that is where certain amount of cytoplasm is pinched off to create the platelets. So first, before it gets to that point, it has to develop. So megakaryocytes go through a polyploid generation by reduplicating DNA or going through DNA synthesis without any cytokinesis. So after DNA synthesis is complete, cytoplasmic maturation continues with the formation of granules, demarcation and membrane system, and fragmentation of cytoplasm, which actually generates the platelets. You can see that over on the right side of this. So you can have cells which are up to about 64N. So these are just really huge, and they have very large polyploid nuclear areas. Now let's talk about granulocytopoiesis. The important regulators here are GM-CSF and G-CSF. Each granulocyte type has a unique progenitor, and maturation is characterized by loss of what we call the primary granules and accumulation of a set of secondary granules, which we call specific granules. It's also accompanied by at least some lobulation of the nucleus. Sometimes they're bilobed, sometimes they have 3 to 5 lobes. Just depends on the cell type, and we'll look at all of those. So newly formed neutrophils marginate in blood circulation and enter connective tissue as needed. So let's talk about the process of granulocytopoiesis. The first cell that you're going to have to ID here is a very large cell called a promyelocyte. It's characterized by having a very large oval nucleus, homogeneous chromatin, and dark or purple to blue kind of color primary granules in the cytoplasm. And these are often clustered towards the plasma membrane, as you can see in this image. A little bit over here, you can see some of them clustered in these areas. And that's typical of a promyelocyte. It's probably, other than a megakaryocyte, one of the biggest cells that you're going to see. Megakaryocytes don't really come out very nice usually in a bone marrow smear, so often promyelocytes are the largest thing you're going to find there. So the next stage, which is called myelocyte stage, is characterized by the loss of the primary granules and the production of those specific secondary granules. And at this stage, it's committed to becoming one of 3 things. It'll either become a neutrophil, an eosinophil, or a basophil. So when you lose those primary granules, you're going to see the specific granules of either dark red for an eosinophil, dark blue for a basophil, or the smaller neutral looking ones for a neutrophil. And those will be present at the myelocyte stage of development. So this is characterized by being slightly smaller than the promyelocyte. It has an oval nucleus, which tends to be what we call eccentric, off-center. Homogeneous chromatin, and of course the primary granules specific to whichever cell line. Here I believe you're looking at a neutrophilic one, neutrophilic myelocyte. As this process continues and it continues to develop, it becomes what we call a metamyelocyte, and that nucleus becomes slightly indented in a U or V shaped, and then progresses even further, and it's slightly smaller, into like a U or S shape, and at that stage it's called a band cell. So at this stage we have a horseshoe-shaped nucleus, condensed chromatin, and the primary granules are not really visible, and the specific granules are very well developed by this point.